All right. So I'm very excited to have Mary Gardiner with us this morning. Mary is a professor in the OSU Department of Entomology. And when I was a county extension educator in the Akron-Canton area, Mary was a new faculty member. And really from the beginning, she developed community science projects. Many of you may have heard of or participated in the Buckeye Lady Beetle Blitz community science project, where she had folks out there in their gardens with sticky cards, capturing beetles and learning to identify those to get a sense of where the lady beetles are and who they are across the state. And then through the years has developed other pollinator related community science projects like Pollination Investigators, which is a 4-H community science project, and other on the ground community science projects related to pollinators and pollinator health. So really has a lot of experience in that community science arena. She also, the bottom left image is some of the pocket prairies. You may have heard about Mary Gardiner's really exciting research, a 5-year grant that ended up recently in Cleveland, studying vacant properties, looking at different treatments of mowing or not mowing, different planting of native perennials, and then studying what the ecosystem effects of those treatments were. So some really fascinating research, some great conclusions from that, and really looking, kind of shining a light on those Rust Belt cities and some of the opportunities for building ecosystems and encouraging pollinators and others in those urban areas. She's also an author. You may have heard her here on our webinar series, talking about beneficial insects, predators, with her book, Good Garden Bugs. So Mary, thank you so much. I'm excited to have you here this morning. I'm going to stop sharing my screen and turn the virtual podium over to you. Thanks so much for being with us.
Thanks, Denise. Thanks for putting together this awesome series and for inviting me. I'm pleased to be here with such a large group today. Thank you all for joining us. So I'll go ahead and share my screen. So I know you've received a bunch of different presentations as part of this series. I'm going to focus on sort of the broader view of community science, what we've achieved as a group across the globe, what are some of the exciting things that we've learned working through community science, and sort of where does the field of community science go from here? What are some of the challenges? Where are we looking to advance? And so the title of my talk, see if I can hide this. There we go. I had to hide the floating menu. The title of my talk is Contributions of Citizen Science to Entomology, Benefits for Nature and People. And I am presenting this in collaboration with my friend, Helen Roy, who's at the Biological Records Center at the Center for Ecology and Hydrology in the UK. Her and I put together this presentation. So a little bit about me. Some of you know me and some of you don't. I'm a professor here at OSU, Ohio State University, as Denise mentioned. And as she mentioned, I study the ecology of urban green spaces. So I study the value of both residential land and also vacant municipal properties for conservation applications. And I approach these types of research through community engagement. I'm very interested in co-creating research questions with communities and addressing problems that people face in their goals of improving habitat quality and providing habitat for insects. And my overarching question in my lab is, how can greening residential and vacant land strengthen communities and support biodiversity? And we address this in both research arenas and also through outreach opportunities. And many of these Denise mentioned, we work in 4-H and with master gardeners and with homeowners and youth and other groups to study insects. And I also am an advisor of PhD and master's students here. This is a lab picture. This is actually a little bit out of date. We had 2 new students join us this fall, but this is our Zoom COVID-19 lab picture. And this is one of the things I really love about my job, is doing research with people that are in training to be scientists. So today is a brand new presentation for me. I had the opportunity to work with Helen on this annual review of entomology article that's going to be coming out in January 2022. So the article is not even out yet. And our title is The Role of Community Science in Entomology. And so I'm going to be giving you some snippets from this article that we put together through our review over took a couple years to put this together. So I'm really looking forward to showing you some of the cool things that we found. So just to start off, I want to start with what is community science. So community, or what was really often referred to more in the past as citizen science, is the involvement of volunteers in scientific endeavors. And what we're really aiming for in this community science field is balancing both rigorous science and education and engagement to create a great experience that yields important data that can be used to address a variety of issues involving insects. And citizen science or community science does have a very long history. So people have been they have been documenting the occurrence of insects and making biological records for centuries. This is not a new phenomenon. So way back when, there were many expert volunteers, people that were not paid, but they had a great knowledge and interest in doing natural history work. And so many people like John Ray, who was around 16 to 1700s, is considered the father of natural history. And this is his picture. And he traveled around Britain noting species that he observed. So there were all sorts of people documenting natural history far before there were professionally paid scientists. And so that's really the onset of citizen science. But it goes back millennia before that. Farmers and hunters have been making natural history observations for centuries. For instance, Chinese volunteers have tracked locust outbreaks, and that's what's in the back of this picture, for over 3,500 years. So people have been doing citizen science a long time. But there have been a rapid escalation in the last couple decades in community science programs, particularly entomology-focused ones. But unfortunately, as we see this growth in community science, we also see many insects in decline. And this is not just a happenstance. It's actually because of the community science data that we've really been able to document some of these declines, because community science data provides insect information over long time scales and over large spatial scales. And both of those things are necessary to understand population change. And so my goal with this presentation today is to highlight some of these important contributions that community scientists have made while working in insect conservation, insect community science programs. So we're going to talk about a variety of different contributions to insect discoveries, invasions, vectors, insect declines and their drivers, challenges of community science, and global future directions. So I'm going to start with insect discoveries. So anytime that you visit a natural history museum, you'll see the contributions of citizen science. Many of the insects collected come from volunteers. And now with the onslaught of some of these very important online recording tools, we are seeing a huge increase in the number of insect reports by community scientists. For instance, in the iNaturalist platform, there are over 1 million records for bees alone. And in the Global Biodiversity Information Facility, which is another worldwide online database, there are over 1 million records of insects. So what have some of these discoveries been using these online tools? So iNaturalist has led to the discovery of new insect species. This stiletto fly, which is a bumblebee mimic, was seen by a professional scientist when just scrolling through iNaturalist looking at stiletto flies. And he thought to himself, I think this is the specimen I found in the British Museum that was really, really damaged. This professor, Sean Witherington, was unable to make a species description based on the specimens that he had. And he saw this fly picture and realized it was the same specimen. So working with that photographer that submitted that picture, he was able to get more photographs and able to make the description and publish a paper about it. He then got really excited about looking for other insects on iNaturalist and also discovered this really cool lacewing with this dark spot on its wing. This was from Malaysia. And after he saw this photograph, he realized, that does not look like any described species. He visited the area with the community scientists and they were able to find more specimens at that same location and also describe that species. So iNaturalist can have major contributions to our knowledge of insect diversity. But it has many other benefits as well. It has a very low threshold for participation. So if someone's new to the study of insects and they happen to see a honeybee in their car and snap a picture, and somebody goes on iNaturalist and talks to them about what they found, that's a great way to engage someone and get them more interested in what else they might find. And as we know, that is kind of the first step in engaging people in conservation-minded behavior. The database also is a great tool for scientists because we can get a lot of natural history information from looking at iNaturalist. For instance, you can connect bees to their preferred plants for forage. You could also look at trophic interactions, such as this bee being eaten by a spider. So you can start to see some of the life history that's going on in the field. They're also an outstanding resource to track declines and invasions. So we know that unfortunately many insects, such as the rusty patch bumblebee, are in decline. So photographs of that species across the country are really important. We also have many non-native bees that have been introduced, like Haleus pictopes, and so we can track the distribution of this alien species using a tool like this. But online databases aren't the only way that community scientists have made new discoveries of insects. There's also some very cool large-scale sampling networks that have gone on across the world, and one of those is the Swedish malaise trap project. This is a malaise trap. It looks like kind of a crazy tent. What happens is insects flow into the trap, and they go up and they get captured in a container that's up by where the man's hand is attaching it there to the malaise trap. So in this project, they established 50 of these traps across the country. This is the most northern sampling location. And volunteers went out and collected the trap contents for 3 years and also helped process the samples in the laboratory, which I thought was very exciting because that's often where community science ends. People collect the data and they ship the data off to the scientists, and then they don't see it until there's results. And I love that this project brought people into the lab to actually help sort that trap catch. Now, they've only processed 1% so far. This is a new project, but they've already discovered almost 700 new species. And that's incredible. And this isn't in the tropics where we think, oh my gosh, there's so many undiscovered species. This is in Sweden. It's a temperate climate. So I think that's really, really exciting. There are also opportunities for community science discoveries through ecotourism. This is an example from a faculty member in Peru who runs a community science program at a lodge called the Refugio Amazonas Lodge in Peru. He sets up blacklight traps and allows the guests at the lodge to come out and help him catalog the insects that come to these blacklights. And these efforts have yielded 4 new species of the arctiny moths, of these very pretty arctiny moths. And so this is another opportunity that we could think about doing more of, I think, here in the US with campgrounds and natural areas and things. I know a lot of metro parks do community science, but I think there's even more ways that we could engage visitors in sort of one-time opportunities to engage in community science with real impacts such as this. Oops. Community scientists have also led to the rediscovery of insects. Lazarus species are species that were rediscovered after we thought they had gone extinct. One notable example is the 9-spotted lady beetle. This species was discovered as part of the Lost Ladybug project, which was ran out of Cornell University. And a community scientist discovered this beetle after it had not been seen in 14 years. Another example comes from Chile, where a citizen scientist found this thick-headed fly that had not been seen in almost 50 years. And the collection of the fly prompted research into its life history and conservation. So several papers were published talking about where it lived, what it ate, what kinds of habitat needed to be conserved to protect it. And so these discoveries can have real meaningful impacts for conservation. Community science has also led to the study of many important biological invasions that could not be studied using traditional scientific methods. When biological invasions happen, they start off in a very small area. And so it's often a volunteer who finds this invader and makes the first report. So that's one very important way that communities engage in the study of invasive species. Also, as these species spread, you need a ton of people on the ground collecting data to see how fast, where are they going, how quickly are they moving. And we just don't have the capacity as researchers to do that sort of thing on our own. And so this is a very, very important area for this, the contributions of community scientists. And within the insect world, there are several major invasive pests that have been aided by the study. Our understanding of these pests have been aided by community science. Two that I really wanted to highlight are the Harlequin lady beetle, which is shown here in the Guardian newspaper, tracking its incredible expansion across the UK. This species is also exceedingly abundant here in the US, and I'm going to talk about that. And the brown marmorated stink bug. There are some great community science programs focused on this very damaging agricultural pest, which is also a nuisance to homeowners. Here you can see this was in the New York Times. Move over bed bugs, stink bugs have landed, and these folks are trying to remove big piles of these insects from their front porch. So both of these insects have received a great deal of study with citizen science. I'm going to focus on a case study of the Harlequin lady beetle as a biological invader and give you some examples of just how in-depth of a knowledge we can gain by working with communities. So first, a little bit of background on this insect. So the Harlequin lady beetle is native to Asia, but it's now found on all continents except for Antarctica. So it's spread all across the world. And community scientists have tracked the spread of this insect across Europe, South America, and the US. And this large publication with 1 million authors shows you these are people from all over the world who all run different community science programs or data collection programs focused on the Harlequin lady beetle. So this insect is having a global impact on biodiversity. And not only is it present in all these areas, but it can become very abundant very quickly, taking over habitat and niche space that was occupied by native lady beetle species. This is a picture I took in Chile in 2013 in an alfalfa field. And the Harlequin lady beetle was first found in Chile in 2008. So this was just a couple years later in 2013. And just in this little patch, I found 7 larvae and pupae of this insect. So it can be incredibly abundant. And when I came to Ohio State, as Denise mentioned, I knew that this insect was very abundant in the Midwestern US. But a survey of Ohio's lady beetle fauna to determine how severe the invasion had been and what the status of native lady beetle populations currently was had not been done. And so I started the Buckeye Lady Beetle Blitz volunteer network to answer those questions. And community scientists tracked this Harlequin lady beetle and native species. And out of this effort, we developed a large multi-year data set, have published 3 papers, trained multiple students, and developed information needed to act from a conservation standpoint. So I'm going to give you a little bit of information about what we did as part of the Buckeye Lady Beetle Blitz. So lady beetles are very charismatic. Even if people hate insects, they usually have a fondness or at least acceptance of lady beetles because they're very, very pretty. And because they're very charismatic and also very economically important, these are predators of some of our biggest agricultural pests like aphids and scales, people have been studying them for a long time. You can go back into the historical record and find data on communities of lady beetles in Ohio in the 1800s. So this is from 1879 in the Society of Natural History Journal from Cincinnati. And if you look in this publication, it lists a number of different species of ladybugs that were found in Ohio. And back then we had Hippodamia convergens, which is this is a drawing of Hippodamia convergens, the 9-spotted ladybug, and the 2-spotted ladybug in the records. So that was about 140 years ago. And if we fast forward 100 years, this is a paper also from Ohio. This is from Bowling Green, Ohio. this group went out and surveyed ladybugs on milkweeds. And again, they find Hippodamia convergens, Adalia bipunctata, the 2-spotted lady beetle, and Coccinella novumnota, the 9-spotted lady beetle. So all of this time, the same species of lady beetles are being found in Ohio. But then all of a sudden, as we start to see populations of alien species, including the Harlequin lady beetle occurring in Ohio and other parts of the US, we start to see these species disappearing. So there have been publications talking about the rarity of the 9-spotted lady beetle, how the Hippodamia convergens, the convergent ladybug, is disappearing, and also the rarity of this 2-spotted lady beetle. And in fact, even as a lady beetle researcher, I have only seen these a handful of times in nature in my career, even though I'm always looking for them. So the Buckeye lady beetle Blitz, which ran from 2009 to 14, aimed at determining the abundance of different lady beetles across Ohio in residential gardens. And our idea was to compare these historical communities found in mosquito records, museum records, to current day communities. And we also wanted to identify habitats that support native lady beetles. So I recruited participants across the state who were given these toolkits that included yellow sticky card traps, an identification and instruction card, and data sheets. They collected their data and sent it back to us to verify. And we held programs across the state to train folks in how to do this. So these are our sampling sites across the years. You can see that we had a large number of participants. The total number of gardens surveyed was 349. And here are 2 kids with their yellow sticky trap. These stars are major cities where we held workshops. You can kind of see we ended up with a transect going across the state for the most part, from Cincinnati through Columbus up to the city of Cleveland. So what did people do? Well, people picked a place in their home garden to sample for ladybugs. And they put out this yellow sticky card, which is a bright yellow card with an adhesive. I know I'm going to get this question. Why did you collect the ladybugs instead of just observing them? Well, for 2 reasons. And I'm going to show one of those reasons at the end. One is, unfortunately, it's very challenging for community scientists to identify ladybugs. Number 2, the sticky cards are designed to collect a very small number of beetles. And so we're really looking here at presence absence as a way of tracking whether populations of those species occur in the region. These are not capturing hundreds of beetles per card. So they're a very low impact, highly accurate tool to track lady beetle communities. So people put out these cards, left them there for 7 days, took them back into their house. And then we wanted to see first, can people identify the lady beetles? And so we had them look at the trap and tell us on the trap, what do they think they found and where is it on the trap? So later we could go back and see how accurate they are in their identification. They then sent us the trap and their data sheet, and we were able to check it and record the lady beetles that they found. So we got a whole bunch of people sending back their traps. So one thing that was great about this program is that about 80% of the people I gave toolkits to sent back the toolkits. So I think one of the reasons for that was it was not a lot of work to do the project. You could do it at home. And we were very clear about how we were going to use the information and why it was important. And I think those are really critical if you're designing a program like this. So we got all of the toolkits back from people. And then I worked with graduate students and undergraduates to check each card and verify the volunteers' identifications. Along the same time, we visited 30 museums across the US to see what sorts of records of beetles these museums had for Ohio. And this involved personal visits, but also help from various curators who checked their beetle collections and provided me with that data. We also looked in online databases. Some museums actually post their beetle faunas online, so you can do that. And we recorded all Ohio beetle records from those museums from 1900 to 2010 so that we could compare those data with the beetle data that we were getting from our survey. So what did we find? Well, first, our Buckeye Lady Beetle Blitz volunteers confirmed that alien species, including the Harlequin lady beetle, dominate the lady beetle fauna in Ohio. This is the Harlequin lady beetle, Harmonia axorides. We found 333 of these beetles. You can see that that's among the most abundant species. The other non-native species that we found are in bold. Unfortunately, because all of these aliens feed on aphids, the native lady beetles that also feed on aphids were rare. And those that remained fed mostly on fungus or scales. Now, we also wanted to compare the data that we found in the Buckeye Lady Beetle Blitz to museum data. And what we found was a major shift in the community. So if you look at this yellow box represents the time period in Ohio when there were not exotic lady beetles present, from 1900 through the 60s. And during this time, there were a variety of native ladybugs present, and they occupied 100% of the community. So that's these 4 species. The 3 here, individual, the 2 spotted, the 9 spotted, and the convergent, and then all others. So that occupied the entire community. Then, starting in the 70s, we had non-native species. And by the mid-2000s, we started to have a community dominated by non-native species. And this gray bar here is just the contribution of the Harlequin lady beetle. So it is now about 40% of the lady beetle community, whereas native species are in total just 20%. So why do we think this happened? Well, because we're seeing this decline in aphid feeding species, we believe that there's a competition for food is driving this change. So before there were exotic species, native species could feed on all of the aphids that were present. But then along came the Harlequin lady beetle, and it was a superior competitor, and it started eating so many aphids that there was less prey available for the natives. So we also wondered, well, where are the native lady beetles? We're still finding some in some people's gardens. Why is this? And we hypothesized that a habitat compression had occurred where native ladybugs were moving out of really aphid-rich habitats like agriculture and only surviving in their ancestral habitats like forests and grasslands that they would have used over evolutionary time in the US. And these habitats are known to not be as dominated by exotics as agricultural habitats. So we tested this by looking at the landscapes surrounding each one of our volunteers' house. And what we found was when a landscape surrounding the volunteers' garden was mostly agricultural, they had more exotic beetles. And when the landscape was dominated by forests and grasslands, they had more native beetles in them. And so from this study, we realized that exotics are a major threat to native ladybugs. And while this isn't an easy solution, the way to conserve native ladybugs is to conserve our native forest and grassland habitat, because this is where they're really thriving. These habitats have fewer exotics. They also provide a critical overwintering habitat. Native ladybugs, many of them, overwinter at the bases of trees and shrubs in the soil. And this is a picture of an aggregation forming in the fall, emerging in the spring. And so preserving these habitats is a critical point that we need to address if we want to conserve native ladybugs. So here you can see the time frame that it took to get this information, the contribution of the people to getting the data, the importance of the historical record, and how we should continue to make contributions to that record now to museums, and what sorts of information can come of it. Now, this is only one of many, many types of contributions to the study of invasives that occur, and I wanted to highlight another one. Many of you, especially during COVID, I feel like we were inundated with studies and news articles about murder hornets. So the Asian hornet is an invasive species that is spreading across the world. And although you might not think of this as a major insect that you want to study as a community scientist, community science has played a critical role in eradicating the murder hornet. So not only can people study the spread of something like the Harlequin lady beetle, their data can actually prevent these damaging effects of alien species from even happening by pinpointing these initial aggregations or initial populations. So this is the case with the murder hornet. So the murder hornet has been found in the UK every year since 2016 by community scientists. And they have developed an online reporting scheme, and this is their smartphone app that's used by thousands of people. And if they think they find a murder hornet, they take a picture of it and they send it in. And this has led to the annual detection and eradication of nests of the Asian hornet. And predictive models looking at the habitat and the temperature and everything have shown that, if people had not done this, that hornet would be present across many areas of the UK. And so these eradications have prevented it from occurring in the area and are really, really important because this hornet can have really damaging impacts. Now, we know here in the US that we are also having some reports of Asian hornets, especially, so far just in Washington State, thankfully. And the USDA and researchers there have really been on top of this issue. And they have also established a citizen science program. And beekeepers have played a critical role in documenting the occurrence of these hornets and getting researchers the information they need to eradicate them. So their program focuses on early detection and faster eradication. And they have a trapping program that shows you how to put out monitoring traps for the Asian hornet. And this is a picture of a group of researchers coming in following a community scientist report to remove a nest. So they act very quickly to get rid of these. Community science has also played a critical role in vector ecology and preventing disease spread from vector transmitted pathogens. There are many vector surveillance programs that rely solely on community science that track things like mosquitoes, hissing bugs, and ticks. Some of these have you submit photographs or maybe the specimen or submit a report if you've been bitten by the insect. And they use that information to look at where these different vectors are occurring, what people's risks are of being bitten, and so forth. An example of this is a project that occurred in the Netherlands where 50,000 tick bite reports were submitted to a community science program. And they use that to generate a hazard map. And this is the hazard map. So it shows you if you're out hiking, where are you more likely to encounter a tick and be bitten? And you can see as you move north, you're more likely to be bitten by a tick. And so if people are going out in these areas, these sorts of data are very useful in understanding your risk. These kind of long-term data and highly abundant data can also provide important information on changes that occur within vector distributions. So there was a 60-year study that occurred in Finland. And in this study, people provided the population of ticks, whether or not they found them across 60 years. And what they found was these certain tick species were moving northward over the 60-year period. So in certain years, for quite some time, people in northern Finland did not encounter ticks. And then slowly but surely, the ticks started moving north. And climate change was identified as a potential driver of the movement of these ticks. And in addition to climate change, community science can address many different drivers of biodiversity change. So things like urbanization or pollution are also tracked by community scientists. But I wanted to show a couple more phenology changes just because there's some amazing data in this area. So when people collect data on when things occur year after year after year, we can start to see whether or not those occurrences change over time. So programs like the UK Nature's Calendar have been recording when different plants bloom, when different insects are active, and so forth for over 300 years. This is an incredible contribution of community scientists. And these data have been used to study a large number of different species, both plants and insects. One study found over 260 moths have declined across Ireland, whereas 160 have increased over this 300 years. Now, why is that? Well, they believe that the patterns are linked to climate change because those that are decreasing are the cold adapted northern species, whereas those that are increasing are more southern, warm adapted species. So this is illustrating a shift in the moth fauna that there's no way we could understand without the contributions of community science. Community science is also moving from conservation to action, which is a really important frontier for the field. So some examples of this are gardening for wildlife programs that also have you record some data on the impact of your garden. And an example of this is a program that had people install drought-tolerant vegetation in LA and Los Angeles, California. And what people found was that when they did this and they tracked the insects in their yard, they found a greater insect species richness in these green spaces than in more traditional lawn landscapes that were dominated by irrigated turf grass and non-native ornamental plants. In addition to monitoring, people have also changed their management as part of community scientist programs. And a great example of that is the No Mow May campaign. This campaign started in Wisconsin by a professor who was interested in providing forage for bees in the early season. We know that dandelions are really abundant in lawns early season. Typically, people mow them. Those dandelions are there before there's a lot of other forage available for bees. And so the idea was in Wisconsin to not mow your lawn in May, and that would provide this forage before other types of forage were available. And so they did a large campaign about this, and people really were excited, and they had tremendous participation. And now No Mow May has really spread across a lot of the northern US where dandelions tend to occur in April and May. Now, citizen science has led to many, many advances in the study of insects, but it is not without its challenges. And especially when we're talking about entomofauna, there are some real roadblocks sometimes. Insects are small and they're hard to identify. And if you walked into my lab, you would see massive microscopes and people, being able to look at these things at many times their magnification. And so when you're asking people to make identifications in the field without that resource, it's hard. And so because of that, people tend to report certain kinds of insects, ones that are larger, things that are more charismatic or more distinctive, and also things that are more rare tend to get overreported. And these lead to biases in the data. And it can also make it challenging to interpret the data, to trust the data that you get. And even if the data are accurate, it can be perceived as inaccurate by peer reviewers, which can make it a challenge for us to publish the data on behalf of our community scientists. And that's really a struggle because then people are not going to get the information and we're not going to advance the field of entomology. Now, these are some insects here in this picture that we could probably easily teach folks to identify. But the struggle is when you really get down to brass tacks, a lot of times what you're looking at are a bunch of small things that really look the same. And so, it is hard. And so this is something as scientific researchers running these programs, we really need to think carefully about what insects are appropriate for this kind of study and how are we going to collect our data. And I mentioned in my community science program, Buckeye Lady Beetle Blitz, that we used the sticky cards so that we could provide verification. And this was based on the challenge that people have distinguishing ladybugs. And I wanted to just show you some of these data to illustrate this. So these are data from that program that we actually analyzed to see whether or not people could be relied on to make their own identifications. Like maybe we could just have people go out to their garden and survey without trapping. So what you're seeing in this graph are all of these different ladybugs that people tracked. And then there are two colored bars. The dark bars are the direct citizen science data. So this is data that people collected themselves. These are the identifications that they made. The verified are the ones that I checked and identified. So these are the actual beetles that people collected. And what you will see here in this yellow box, these are rare species. And what you see is we see more reports of rare species in these dark bars by the volunteers than actually were there. So for instance, this Hippodamia convergens, this convergent lady beetle, is known to be very rare in Ohio. People thought they found it quite a bit, but they actually didn't. People also thought they found this Hippodamia tremdesa punctata a fair amount. They didn't find any of those. So if I would have taken these direct data without checking them, we would have overestimated how well native species were doing. And we believe this is due to a conservation bias. Everybody hopes to find rare things, right? We hope our garden supports rare stuff. But the problem is when people provide these sorts of data, it's not accurate. And so we're making the wrong conclusions about the information. And we found also interesting things for the exotic species. So these are the 4 exotic species tracked by the program. People tended to underreport exotic ladybugs or alien ladybugs. And so here you can see, for instance, for this Harlequin ladybug, my count was much higher than the reported count from the community scientists. And this is also part of conservation bias. We hope to find rare species, not alien species thriving in our landscape, even though alien species are found in almost exactly the same sorts of managed habitat niches as natives, and it really has nothing to do with your garden management practices. So what do we do as community science practitioners to prevent or mitigate these issues? Well, there's a lot of approaches that you can do. I mean, we can revert back to our own traditional methods of collecting our own data, but as I've mentioned throughout this presentation, that's often not a good way to go. It's not going to provide the information we need. And in some cases, direct data provided directly from volunteers is perfectly fine. It works great. So if you're just having someone count one type of insect, maybe an invasive pest like the brown marmorated stink bug, and they're unlikely to confuse it with something else, that can work really well. But oftentimes we fall into this middle ground where some type of verification is present. And that kind of affects things like the cost. So as we have in direct science, citizen science, the lowest cost, as we move to verified, that's going to increase the cost, but it's also going to increase the accuracy. But it's also going to slow down the dissemination of the findings. And so you saw those piles in my lab. You know, if we relied on the people submitting the ladybug data, we could have just published it right away on the website. Instead, they sat there for months as I had time and the students had time to count them. So there's this delay. So depending on the urgency of the issue, for instance, the Asian hornet, you would want people to submit if they think they found one and provide a photograph, and then you could go and get it if it was the correct one. You wouldn't want to have samples shipped in that would sit there because it's too urgent. This was an important issue, though, because we did not want to overestimate native species. We needed to know the exact population. So this was a good way for us to do that. But all of these issues influence program costs, the number of volunteers that you can have, the speed you can get your information out. And so as a program manager, these are important things to think about if you're going to develop a program like this. There are also some other important future directions for community science that I see as critical to us continuing to see this area as a major contributor to conservation and the study of insects in general. So we must address our geographic coverage for one thing. If you look at the number of published community science programs, you can see that there is a major geographic bias with the US, the UK, and European and Australian countries in the top 10. And the US and the UK really leading with the number of published studies. And so this is leaving out some really critical habitat for insects, like high altitude areas and tropical areas where many insects live. And we don't know a lot about them, how they're doing. There might be some important vector questions to ask in some of these habitats, and we're not able to do that through community science currently, and that needs to be an area of a lot of growth. There's also a taxonomic bias in the types of insects people are studying. So not surprisingly, people tend to focus community science programs on insects we think people might like, or insects we know people understand the harm from, like pests or vectors. And so you tend to see things about butterflies and bees and beetles being popular. And if they are about things that are kind of creepy, they're about things that might harm us or harm our crops. And so there is a real need for scientists to think of past this sort of idea and to give people a chance to contribute to a wider range of different insect studies. Because when people know more about why this is important, they really are willing to engage. And I wanted to show just some extreme examples of this. So this first one here is a baboon spider, which occurs in Africa. It's a big tarantula. And so you would think, no, like no one's going to want to do this. But when the researchers talked to people about the threat that this spider group was facing, that it was being illegally harvested to be a pet, and also that it was losing its habitat, people across different countries, many hundreds of people went out and took photographs of these. And they were able to develop range maps to figure out where to protect them. So people are willing to participate if you illustrate to them why it's important. And even in a creepier example, absolutely love this program called Report a Worm. So these scientists work on these type of worms, these big worms that spend most of their life living inside insects. And the worm is able to control the insect's brain. So when the worm is ready to leave the insect, reproduce, and its offspring lay eggs, they somehow are able to change the insect's behavior so it flies to water and lands in water. And when that happens, the worm emerges. Well, in urban areas, these worms emerge in people's sinks and toilets, in dog water bowls, and all sorts of habitats where you might have water around your house. And so these researchers thought about it and said, you know, I think people would be horrified if they found this thing, and they will go to Google and they will say, what is this giant worm in my dog's water dish? And so what they did was they made their website so that when people searched various things like large worm in toilet, this website would come up and it said, report a worm. Did you just find a giant worm in your dog's water dish? Send it to us. And the reason they wanted these is these people specialized in the taxonomy of these worms, but they would go out all over the place and could not find them. And so they were unable to make progress. Well, once they had these data from the public, they started to be able to put the pieces together in their phylogeny of these worms and make some real progress in understanding their relatedness and how many species there were and things like that. So this is a picture that they received from someone of one of the worms in a dog water dish. And here's the beetle that it was living within. So people sent these to them. Another area that we really need to focus on is increasing the types of opportunities for people so that a greater diversity of people can participate in citizen science. Right now, community scientists rarely represent the diversity of people that live where a program takes place. And in one study across 7,000 programs, the people participating averaged 94% white and 73% college educated. So that's leaving out a great proportion of people that could enjoy and benefit from and contribute to community science. Historically, groups that have been underrepresented in science are also underrepresented in community science. And that means that those groups aren't able to influence the questions, the methods, they're not able to reap the benefits, and they're unlikely to be able to be motivated to seek a career in science through participation. And we know that community science for youth is a really important portal to community, to engagement in scientific careers. So how do we do this? Well, I don't have all the answers here, but I just think that this is really, really important. So the number one thing to think about is that co-creation of the project is critical. Instead of coming up with a project and going to people and saying, I want to study this, here's the information, what we really need to be doing is going to people and saying, what are your priorities for your environment? What questions do you have? What concerns do you have about the environment that you're living in biodiversity and the natural world? And when people come to you with those questions, we can start to conceive of a project that they would be very much interested in. If people are not interested, they don't see it as important, they're not going to engage in the project. Also, it's important to understand how people want to receive the information, how they want to engage, and what barriers they might have. They might be time, financial, mobility. There's a lot of different barriers that people could face that could influence their participation. So where do we start? Well, if we engage people from the onset, we can have them help as a collective to make sure that we make a program as inclusive as possible. So people can advise researchers on how to get the word out. In our community, how is the best way to reach people? What sort of language barriers might there be? And could some of the community members engaged in project development help overcome those by translating materials? Will the use of technology aid participation or deter it? In certain parts of the world, technology can be a barrier. In other parts, it can be a great facilitator. It's important to gauge that from your community before you design a project. Time and effort. If you design a program that takes 20 hours a week to complete, Do you think that working people are going to participate? No. If people have to hike 10 miles, will people with mobility problems or limitations be able to participate? No. So we can think also about ways that you can engage people in various levels of detail that might vary in the time commitment, how much you have to get out in the field, things like this. And also a big thing in the scientific community now is, should we be compensating people for participating? It's a lot of work in some of these programs. Should we be paying community scientists? So that's another issue that's being discussed and really thought about a lot. So just to finish up here, how do we move forward? So I was really excited to hear that the next presentation from the Xerces Society is about translating from monitoring to action, because I also think that that's a key area of growth for community scientists. So clearly from this presentation, I hope I've shown just the sheer amount of data that's been generated by community scientists studying all sorts of different insects and other arthropods. Everything from charismatic conservation targets to report a worm. So people are interested. So we have a willing group of community scientists. If we were able to, as a global scientific community, start to standardize some of our protocols, we would have a lot better luck doing things like risk mapping, targeting conservation interventions, understanding the spread of invaders, if we all were able to use the same community science protocols. And so there's been a push for more standardization. And I want to use an example of pollinators here to just illustrate this. So as you all probably know, there are many programs across the world that study pollinators. And these pollinators focus on things like just opportunistic sightings. They might involve structured observations or even in-depth surveys. And any number of different reasons for doing these surveys or reporting these observations might occur, such as just understanding distributions or monitoring pollination services. So just some examples here. For instance, this bumblebee watch is tracking conservation of bumblebees across North America. And there's a similar program, Save Our Bumblebee, in Chile. If both of these programs started using exactly the same methods to track bumblebees, you could start to compare gains and losses, what kinds of habitats are affecting these species across a broader area, and it could spread, to other parts of the world. Here in the structured observations, many of you are probably familiar with this awesome Great Sunflower Project that's run in the US. This involves flower counts at sunflowers of pollinators, and also looking at pollination services, how many seeds are produced. Again, this sort of thing could be replicated across the world to look at pollination services. So if we could get some standardization, it would really be an awesome way to work together to conserve pollinators and pollination services. So just to wrap up, I want to thank all of you for being here and for all of your work as community scientists. I want to thank Denise for organizing this and highlight my funding from the Integrated Pest Management Program here at Ohio State. So thank you very much. And if there's time, I will address some questions.
All right, great. Thanks, Mary. Thanks for a really comprehensive overview. And we visited lots of different insect orders, which I think is helpful, can take us out of what we're knowledgeable about and generate different questions and ideas. So one of the questions that came up was if you're listing observations on something like iNaturalist, how is that data shared? And I thought maybe you could just talk briefly about GBIF or other ways that researchers find data.
Yeah, so we, through iNaturalist, we can download data sets from that website. Depending on the question, we might use verified sets or all the data, depending on what we're looking at. But a lot of what researchers use in iNaturalist, they search the database and collect their own data from the database. For instance, if you want to know all of the time someone found a bumblebee on a particular type of plant, you're going through the database and sorting it by, maybe region and species, and then just going photo by photo. So there's a lot of sorting that happens when you're using that data as well. But yes, there are summaries that you can access that provide the data from the website.
Great. I think sometimes folks worry that, well, I took this picture, but does it really matter? But it does go into a larger data set that researchers can with their
the data in iNaturalist are used frequently. in many published studies. And so I just highlighted a couple of examples, but yes, those data are used frequently.
There were a number of questions about your Buckeye Lady Beetle Blitz project, and one of them was about the Harlequin beetle and other invasive beetles, how they spread across the globe.
So it's unknown how the Harlequin lady beetle was introduced into the US, but historically it was actually purposefully introduced as a pest management in crops. So before there was a lot of knowledge about the potential negative impacts of this, the thought was, well, these beetles will naturally control pests and it will decrease the use of pesticides. And so they were being collected and redistributed as a means of pest control. But the actual populations that we have here, it's unclear how they were established and actually thought to be accidental introductions. And that can happen through a variety of ways, often on plant material, when plants are redistributed across the world, or in shipments of other fruits or vegetables or other agricultural products, insects can be introduced.
Okay, and are the sales of ladybugs problematic?
The sales of ladybugs, it's unclear if they're problematic from a conservation standpoint, but it is possible. So that you can purchase you can purchase Hippodamia convergens, the convergent ladybug. And when you purchase those ladybugs, they are wild collected. So people go out to mountainous areas in California and dig them from their overwintering grounds and sell them. And so to me, that's harming them in their natural habitat in large numbers and shipping them around the country. And so right there, I don't know that it's necessarily having a negative impact on their populations in California. I have not read a paper that says that, but just going out to their natural habitats and collecting them, you should know that that's what you're getting. They're not, that particular type of insect is not reared in the lab. Because of that, it could also have diseases or pests within it, like parasitoids. And if it's introducing new pathogens to new parts of the country or world when they're re-released, even if that insect is native in that area, it could introduce a novel pathogen. Again, we don't know that that has occurred, but it is a possibility. The other reason I would not release them is that they really don't do you any good. So what happens when you release those, they're shipped to you in a diapause state, and because they were taken from their overwintering habitat. And when they come, active, their first instinct is to fly down into the Central Valley. So they're going to leave your yard. So you're not going to benefit them from pest control. The best thing to do is to make your yard a great place for beneficial insects to live by planting lots of flowers, by not spraying pesticides, by allowing weeds to thrive in certain areas where you can tolerate them, because those are also food. A bottom-up approach is far better than buying the ladybugs.
You kind of mentioned this with your yellow sticky traps. There was a question about bycatch and what bycatch is and whether you use the bycatch. But then a more general question, you mentioned this about beetles and over-collecting, but the question was about bees in studies, and is there a concern about over-collecting bees?
So bycatch is a bycatch means insects that were caught by happenstance that were not the focus of the study. I care deeply about making use of all data that we collect and focus a great deal of effort to use bycatch. In fact, many of my grad students' first paper is on bycatch. And what we do is, say we're doing a project, focus on spiders, and in those traps, we also collect ground beetles. Well, we also already have all the other data that was in that spider paper, the landscape data, the local habitat data, the questions, etc. And so often when people are first learning how to write a publication, I will have them sort some of the bycatch and use all of the other data we've already collected to look at how those data might influence another insect. And so I make a major effort to use that bycatch. And we've found some very interesting things. Bycatch is not without value by any means. You can never use everything, but we make a real effort to use as much of it as possible so that we're not just killing things. We also make an effort to only sample as much as we need to. We don't sample constantly. We collect just the amount of data we need. And I think most ecologists you're going to see do this. With bee samples, I have not known of any evidence that the amount of bee sampling that occurs is harming the bee community. There are not a great number of people that do this kind of sampling where I would expect, I mean, there are a lot of bee scientists, but it's not like every state has 100 bee scientists out there collecting bees. So the number of bees that are being collected is incredibly small. The other thing is the traps that we use to collect bees are tiny little pans that are left out for 48 hours. And the idea, just like the sticky cards, is that you collect a small number of the bees so that you can understand the population more generally. But unfortunately with bees, they are extremely hard to identify, and we need the actual specimen as our record. In fact, I take all my bees, even though I've been doing this for a long time, to Sam Droege, who I think was a previous presenter, and he checks them. So that's how hard, I mean, some of these things are so challenging. I fly a big box of all my specimens to DC with me every year, visit him, and he's nice enough to go through them. So there are not a tremendous amount of people who do this, and even fewer people who can identify these bees to species, even with the specimens.
Great. David mentions the Oregon Bee Project as a kind of a gold standard for community science. And there were other questions about where can folks find out about community science projects? Is there a place on iNaturalist they can search, or is there a clearinghouse?
Oh my gosh, that's a good question. So there is a couple websites that catalogs community science programs, but not all programs are going to be on those sites. I think if you're interested in a local program where you can have a lot of engagement with the researchers, I would look at my state university or public university in my country, or school that studies, the taxa that you want to study, whether that's insects or birds or what have you. And if you contact some ecologists at that university or at a big park system in your area, they're probably going to be able to identify the best options and opportunities for you to engage. And actually, I just recently got to review that Oregon Bee Project. It is an awesome project. The person that runs that is outstanding.
Great. Folks, so it is just a little after the hour. If you need to hop off, feel free to do that. I'm sure Mary would appreciate it. Thank you. I've already put that in the chat box. And then we'll continue to answer some questions and keep the recording on so you can come back. Let's see. Sorry, Mary, I usually have a question just ready to go. So someone had a comment about those volunteers that have their specimens verified, and how perhaps those volunteers can be moved into different roles as they learn improve their skills with identification.
So I think there's a lot of opportunity to have people take on more responsibility in community science. And I didn't get into this so much today because I feel like that topic is more focused on the practitioners, but I think that we need to allow people more opportunities to do more than collect the data. So people have interest in working in labs and seeing the, behind the scenes aspect. How do you identify these things? What do you do with the bycatch? Can we look at that? Can we identify that? How do we pin the insects? How do you analyze the data? Now, not everyone is going to want to spend hours making ArcGIS maps and doing statistics with us, but some people do. And I would love to, and this is something that I'm really focused on now, my current work, make more opportunities for people who maybe they were, a scientist in their career, now they're retired and they want to engage. Maybe they've never done science, but they're just fascinated by it, want to learn how to pin bees. So thinking about all sorts of different opportunities for people to work in the lab if they want to, and to learn as much as they can is important. And also for people to analyze and publish the data. So there's some real barriers that we face in trying to have community scientists as co-authors. And I think that the scientific community is starting to recognize that and think about ways that we can provide those opportunities too.
Okay. Martha wonders if your sticky cards contain a pheromone, or they just catch by dumb luck.
They don't contain a pheromone. They're actually attracted to the yellow color. So ladybugs are attracted to plants that look unhealthy. And the reason that is because they're looking for aphids, these ladybugs. And when aphids feed on a plant, they suck the juice out of it and the plant kind of turns yellow. And the sticky card is supposed to mimic that colored plant. And so we are trying to really collect specific insects that are looking for aphid damaged plants in this case.
Susan has an interesting question. She has acreage in West Virginia next to a forest area and wants to offer some ecotourism activities, but she's hesitant to start because she's an amateur. She does use iNaturalist. Is that enough for recording and hosting such activities, or are there other resources you can suggest?
Well, I think the first thing would be to maybe figure out a few different things you wanted to highlight in the area. And maybe focus on, those aspects. The other thing that you could do, and this is something I do all the time, if I want to bring a group in, but I don't know anything about the topic yet, I'll set up the event and host an expert. So a lot of times when I organize a program, I might be one speaker, but then I'll have someone come in and talk about, native plant propagation or how to manage your forest habitat for bird species or whatever the topic is. And so you might have the site and the interest and the ability to organize the event, but if you don't yet have the information, that might be a way too that you could engage people by having some local naturalists come out and speak. And people are usually willing to do this. We like doing this kind of stuff, so.
Susan wonders if there is an equivalent of Nature's Calendar in the US. Maybe you can mention the National Phenology Network. And
right. There is a National Phenology Network in the US, and I actually have that in the paper, just due to time, only mention the one. But yes, we do similar. We don't have 300 years, but we do have quite a bit of data in the US too.
Yeah, so some great ways to get involved in phenology. And again, that data is really widely used by researchers. So you're doing a lot when you participate in that community science outreach. Mary, there are a few questions that are going to tap your knowledge about bees and bee habitat. I know that's right up your alley. So let's kind of address some of those. So any thoughts on implications of invasive species and including invasive plants and their effects on bees?
Well, invasive species wise, and I'm just speaking from my own knowledge here, so I'm not saying that this is 100%, but I have not recently read or ever read any papers showing that an invasive bee had displaced a native bee, although in certain areas we certainly see more dominance. For instance, in my work in Cleveland, we find an exotic osmia able to exist very commonly in cities, whereas a native osmia that occupies similar nesting resources cannot as frequently occur. And it also, the native species has a host of parasites where the exotic one does not. So there are complicated environmental variables that influence the distribution of these things. So the exotic might not directly be like eating the native one or somehow hurting it, but because it's more tolerant of habitat degradation and it lacks natural predators or parasitoids, it has an advantage. And that's something you see a lot with invasive species. The reason that they're so abundant is that they escape all that when they move to a new area. They no longer have often their natural controls. And also their sheer ability to invade means that they're probably a species that can tolerate a lot of different types of environment, like cities or degraded habitats. And so they often can, outcompete our native species in that way. Exotic plants, man, you can read every number of things. And I'm not talking about invasive plants here. I'm just talking about non-native versus native plants. In some studies, we see that native bees prefer native plants and that those are really important. In other studies, you see that it's the protein to fat ratio in the nectar or the pollen that affects who's feeding on what. So there's a lot of components to this that make it hard. But I think there's a lot of reasons to plant native plants. They add those plants back in the landscape. Many birds feed on the seeds. They provide, they're huge oftentimes in the Midwestern US. They're tall. So if you leave them there in the fall, they provide great cover for things in winter temperatures. And we know that bees love them. So I can't say for sure that they're better, but they certainly have a lot of value.
And when you say osmia, you're talking mason bees, just to help people kind of translate that.
Oh, I'm sorry. Yes, cavity nesting bees.
For a hillside wildflower garden, is it better for the bees to mow down the dead flowers in the fall or to leave them alone?
Well, I think for many things, it's better to leave them alone. Now, the only thing I can think of in terms of cuttings would be some species of wasps and bees will nest and cut stems. And it's often suggested by experts that I've listened to go out and snip some of the flower heads off to allow that type of nesting. So I tend to just leave everything. My yard looks like a dried down mess right now. It's like got all this stuff just, but you know what? Snow will come and all will be fine. I just leave it because I see things out there all winter long, birds feeding on the seeds. And in the spring, I have a lot of activity and I'll wait until stuff starts to sprout out the bottom and then I'll cut off the tops. That's what I do. But I'm not an expert in garden maintenance in that way.
There's a comment about Bombus pennsylvanicus as possibly being added to the endangered species list. Just wondered if you had any thoughts or any ideas as far as community scientists and helping to document or assist with those kinds of endangered species.
Well, Denise, I know you've got an active bumblebee project, correct? Or you guys have been, you guys have contributed, some of the people in your program have contributed to bumblebee monitoring.
Sure, right.
And so there are many bumblebee monitoring projects out there, and those projects are providing the data that would inform something like whether or not this species would be listed. So I think participating in those is a great idea. I think that there are already enough good bee monitoring programs in the US, at least, that we need to, as I mentioned at the end, I think standardization is a really important thing because it's very hard to take data from all these different programs where people collected stuff differently and try to make sense of it. And so I think as a scientific group of community scientists and people that are paid researchers, we need to come to some decisions about how are we going to monitor things like bumblebees? And can we do this the same across big areas? Because then you can really see what's happening with these different species.
Yeah, great, Mary. And it really brings us back to that U.S. National Native Bee Monitoring Network. You know, they're trying from a research perspective, let alone a community science perspective, to get some standardization together so that we can benefit from each other's work and effort. Mary, I want to thank you for your time this morning and your expertise. There were a few comments in the chat box. I always find you so approachable in your knowledge. You have such depth of knowledge, but you also present that in a way that's really understandable, and it helps us stay motivated about the good that we can do in science, and just a great example of what's happening out there in academia. So again, thank you, Mary.